Hi everyone, my name is Tracy Steinricken and I'm the president of the Australasian Mycological Society. I'm also a postdoc at the CSIRO. Thank you for joining us for our August virtual seminar. Before we begin, I'd like to acknowledge the traditional owners of the lands on which we're all meeting today, here in Australia, in New Zealand and around the world. I'm talking to you from south of Maiwar, which is the Brisbane River in Queensland, and I'm on the traditional homelands of the Yugambeh people. I pay my respects to their elders, past, present, and emerging. Our presenter today is Dr. Kentaro Hosaka. Kentaro is originally from Yokohama, and he did his undergraduate in Japan's southernmost and only subtropical region, Okinawa. His PhD in botany and plant pathology was at Oregon State University in the USA, which was followed by a postdoc at the Chicago Field Museum's Department of Botany. And that was from 2005 to 2008. Since then, he's been the curator of mycology at the Department of Botany at Japan's National Museum of Nature and Science in Tsukuba, where he has been since. Kentaro has several research interests, including the systematics and phylogenetics of mushroom-forming fungi, the biogeography of globally distributed mushrooms, the biodiversity of mushrooms using specimen-based and metagenomic data from soil DNA, and what I consider to be a kind of microbial inception, the biodiversity of symbiotic organisms within mushroom fruiting bodies. So today, Kentaro will be telling us about the natural history of the beautiful blue entolomas of Japan. If you'd like to ask a question or post a comment for him during the talk, you can do so by clicking on the Q&A function at the bottom of your screen. He will answer your questions at the end of the presentation. And then after the discussion, please just take a moment to answer the short survey to help us improve delivery of this seminar series. In the meantime, please help me welcome Dr. Kentaro Hosaka.
All right. Thank you, Tracy, and very kind introduction. And I would also like to thank Bevan and all the organizers for inviting me for this seminar series. It's a great pleasure to talk in front of all of you from my computer in Japan. Although I really want to physically go to Australia soon, under this pandemic, it's going to be very difficult. But hopefully, we all meet physically soon again in Australia. When I was first invited for this seminar series a while ago, I was asked to provide some photos of me, ideally at work. So I chose several photos of me during my fieldwork. But just this morning, I realized that the photo chosen for the AMS webpage is like this. Me holding a papaya at the resort hotel in Hawaii. But seriously, this is a photo of me during the fieldwork in Hawaii several years ago. So this is me during the fieldwork, collecting mushrooms in Hawaii. So today, my talk is titled Natural History of Blue Entolomas from Japan and the World. I have a broad interest in mushroom-forming fungi, and this blue entoloma is not my major research topic. So I have to say that this talk is not very conclusive without many data. So I really would like to introduce you the general patterns of fruiting of blue entoloma in general in Japan and the world. So the outline of talk today is like this. I first begin with the distribution pattern of blue entoloma in Japan, followed by particular fruiting patterns of blue entolomas in the Bonin Islands, which is very south island in Japan. And I'm going to briefly touch on the distribution of blue entoloma from Australia and other parts of the world. And as a final conclusion, although it's not conclusive, the conclusion is the natural history of blue entoloma in Japan and briefly also in Australia. So these are my main research sites in Japan. The Japan mainland is here, and this Tokyo, and this star is actually Tsukuba City, which is my hometown. I'm talking from here. And the Bonin Islands, the subtropical islands, very tiny islands, but very interesting, unique biota, about 1,000 kilometers south from Tokyo. which is oceanic islands, so they have never been connected to major continents. There are also subtropical islands in Japan, which is Okinawa, about 1,600 kilometers southwest from Tokyo, which is continental islands. So they've been connected and disconnected from the mainland China several times. Just brief introduction on the Bonin Islands. It is known to have higher ratio of endemic and endangered taxa for plants and animals. We don't know much about fungal situation, but we have several fungal names that are known only from the Bonin Islands. And one example is that the Japanese red list includes 26 extinct fungal species. Again, we don't know if they are really extinct or not, but those 26 species have never been collected for more than 60 years. And out of those 26 species, 21 are known only from the Bonin Islands. And another important thing is that the Bonin Islands have many endemic, unique plant species, but there are no known native ectomycorrhizal plants. Currently, we have some introduced plants, ectomycorrhizal plants as well, but we have to remember that there are no known native ectomycorrhizal plants there. So this is our main character today, the blue entoloma, Entoloma virescens from the Bonin Island, Japan. There are some lesser known synonyms like Cephalus virescens, Leptonia virescens, but I consistently use Entoloma virescens today. So like you see, it's very beautiful dark blue with part of the yellow discoloration. This is also a very important character. However, the macromorphological character vary. For example, this fruiting body is rather weak and faded to pale blue. This one is very skinny, very skinny stalk, but this yellowish discoloration is very clear. Sometimes it is darker blue, and oftentimes, especially after you pick up those mushrooms and then bring it close to the lab, then you realize that there are so much yellowish discoloration. So they are not beautiful blue anymore, but this is important character for this species. And as a species belonging to the family Entolomataceae, they have pinkish spore mass. So oftentimes when you bring a mushroom to the lab, you realize that there are thick spore prints on the top of the caps and make it very pink. So the important message here is that this species, Entoloma virescens, is blue and they are not tiny. Now, sometimes they are skinny and tiny, like this one. It's about 3 centimeters tall, but the largest one is rather stout looking and more than 10 centimeters tall. So they are not tiny mushrooms. That's important to remember. And in general, this species is considered rare, if not endangered, in many parts of Japan. Now, like the national level red list, we also have regional level red list in each prefecture in Japan. And several prefectures, like here, shown in color, listed the Entoloma virescens as endangered in several different categories, but generally speaking, they're considered rare and endangered in those areas. The other parts of Japan, shown in white, it doesn't mean that the Entoloma virescens is not rare or common in those areas, as they are simply not found in those areas. So the species is certainly rare in many parts of Japan. However, there is one exception, which is the Bonin Islands, which is around here on the map, because Entoloma virescens seems to be very common fruiter in those areas. So this is a similar map, but based on the specimen data. I took those data from the Science Museum Net in Japan, which is like a GBIF integrating all specimen data from all major herbaria and a museum in Japan. So I searched using both Entoloma virescens and also the synonym Entoloma aeruginosa. and found only about 50 records, and distribution patterns very scattered in the central part near Tokyo, central north part, and the western part of Japan, and also it's here in the south. It's from the Bonin Islands. So taking into consideration the both aspects of data based on specimen record and based on regional red list, we can say that the distribution records of Entoloma species is very incomplete. But it could really be reflecting the truth. This species is distributed widely in Japan, but the specific locality is very scattered. And also, there seems to be a vague pattern showing that the western part of Japan have more Entoloma Veraskins fruiting, maybe also more common in the south. So generally speaking, this species seems to be more common in warmer area. So speaking about the warmer area, there are another place in Japan that is subtropical climate besides the Bonin Islands, which is Okinawa, around here, but we have no specimen record. But recently, we received many mushroom specimens from Okinawa because the local museum there couldn't keep the mushroom collection anymore, so they donated to our herbarium in Japan. And among them, I found this specimen with the paper slip with very the bad handwritten characters. This actually is Entoloma Veraskins. I knew it because when I saw the microscope, it had the typical cubic shaped spores, and also it's here is a Japanese name Entoloma Veraskins. And also, I really knew this collection actually. When I opened the box, I quickly realized it because this here, the collector, is actually me. I collected this specimen when I was undergraduate in Okinawa. So I clearly remember that when I collected it, it had very strikingly blue color. But after drying and using the dryer, and it's been kept at the not ideal situation in Okinawan area, the color becomes like this, and it's not beautiful anymore. So my memory is vague, but I kind of remember that it was collected from mixed Castonopsis pinus stands. And this pinus species, Pinus luchuensis, will be very important at the end of my talk. And what about north? We had specimen record from the middle part near Tokyo. However, recently, my friend invited me over to Sendai in Miyagi Prefecture. It's rather north part of Japan. And he showed me around the forest, and I found this Entoloma Veraskins. So based on the current specimen record, it showed only this scattered record. However, we now got specimens from Okinawa, and also we had more recent specimens from Sendai in the Miyagi Prefecture. So it's covering the large area of Japan now. So I'm curious about the northward distribution of this species as well. So here's the Sendai, here's the magnified map, and Sendai is here. And I searched around the specimen record in many areas, but I couldn't find anything from the area, more northern area than Sendai. However, a friend of mine told me that there is a rumor that in the Aomori Prefecture, there is observation records of Entoloma Veraskins. And of course, without specimen record, it's hard to conclude that presence. However, I kind of feel that this species, Entoloma Veraskins, is really distributed in Aomori as well. And what is important is that the ocean in between Hokkaido, which is the northernmost part of Japan, and Honshu, which is the mainland of Japan, there is a line called Blackstone Line, which is a very major biogeographical line separating the distribution of many animal species. For example, one species of bear is only in the south part of Blackstone Line. The other grizzly bear is only in Hokkaido. So my gut feeling is that it also applies to many fungal species. So probably the Blackstone line is functioning as the distribution barrier for many temperate tropical species of mushrooms, including Entoloma veraskins. And I already have some target species for this study, like Entoloma veraskins, and also this very strikingly bright bioluminescent species, Mycena chlorophyllus, seems to be limited in Honshu, mainland of Japan, not in Hokkaido. And also, there are two very gigantic mushrooms with the tropical or subtropical origins. One is Microcephaly gigantea. This photo actually me holding the whole fruit bodies, but this is very gigantic and used to be common only in the very south part of Japan, like Okinawa or Kyushu. But currently, it is quite common around Tokyo as well. So probably this species expanding this distribution northward. The same is true for this Chlorophylla molybditis, the very large poisonous mushrooms, which also has subtropical or tropical origin, but nowadays very common in Tokyo area as well. And we have absolutely no record in Hokkaido, but seems to be some records in the Ibaraki and Miyagi and those areas. So I really think that those 4 species plus many other species of mushrooms are restricted to the south Blackstone line. And I kept explaining about this Entoloma veraskins in Japan. And here's the reason. Among several other blue Entoloma species, Entoloma veraskins has been described first. It is the oldest name among all those other blue Entoloma species. I'm not saying that all other species names are synonyms, like New Zealand species, Hoxteteroid, is probably a very different species from Veraskins. However, when we talk about blue Entoloma taxonomically, it's important that the which comes first. And one other important thing is that the Entoloma veraskins has been described from Japan. So the type locality is Japan and the Bonin Islands. So to understand the taxonomy and the species entity of blue Entoloma, we really need to study the samples from the Bonin Islands, which is type locality. There are some interesting history behind the blue Entoloma and the Bonin Islands, because when the specimen was collected, it was not by a Japanese mycologist, but by a botanist of the U.S. North Pacific Exploring Expedition. This is a huge expedition visiting many places, almost globally, including South America, Southeast Asia, and also part of Japan. And during the time, the islands was inhabited by people from many places in the Pacific, and the Japanese people are not the dominant people in the islands. So the historically is very different. Anyway, in 1854, the first collection of Entoloma veraskins was made from the Bonin Islands by Charles Wright, and then it was later described formally as scientific with the scientific name by Berkeley and Curtis. And since then, because of the unique locality and unique flora and fauna in the Bonin Islands, many Japanese mycologists started investigating the islands for collecting mushrooms. And with the large gap in between, I started collecting mushrooms in the Bonin Islands from 2009. And actually until 2019, I've been visiting the Bonin Islands every year, sometimes 2 or 3 times a year. But because of the pandemic, I couldn't visit the island last year and hope possibly it's not possible this year either. But an important thing is that during my own foray in mushroom in the Bonin Islands, I've collected many specimens of Entoloma veraskins every year, every time. However, it was only from one island in the Bonin Island system. And I'll explain about the island system of the Bonin Islands later on. And the other important thing is that these Japanese mycologists between the early 1900s to early 2000 are all very great, famous, renowned mycologists, like Sanshi Imai, Shio Kobayashi, Tsugo Hongo. They're all great mycologists. And like Imai was specializing in many cup fungi as well. So they can find tiny fruiting bodies as well. However, during this period, absolutely no records of Entoloma veraskins is present. I've searched all their publications during this time. They have not indicated anything about Entoloma veraskins. And I searched Iberia, and no records of Entoloma veraskins from Bonin Islands during this time. So it's very curious. It was originally collected from the Bonin Islands in 1854, and I have collected a lot of specimens of Entoloma veraskins, abundant fruit bodies from Bonin Islands since 2009, but there are no records during this period. So here the map of the Bonin Islands. There are many more islands, but there are two major islands inhabited by humans. One is Chichijima, meaning father island. The other one is Hahajima, mother island. Many smaller islands are also named, like the elder brother island, younger brother island, elder sister island, younger sister island, and many other things. But those are two major islands. And each time when I visit the Bonin Islands for collecting mushrooms, I typically visit both islands. These are separated by about 50 kilometers. And they have shared many plant and animals, although each island has its own endemic tax as well. And throughout my survey since 2009, I visited both islands typically with many other famous great mycologists. And here's my collecting trip number, the total of 20 times, and I have to add more since then, but these are the between 2009 and 2017. And each time, color coded differently. For example, in 2009, I first visited Hahajima, followed by Chichijima. In next year, 2010, started with Chichijima, followed by Hahajima, and so on, for this the duration with those people. So KH is me, Kentaro Hosaka, and there are names of the mycologists. And I don't consider myself as great mycologist, but the other people here are all great. So this is to show that I was not alone. There are more eyes. So we had more chance to find Entoloma veraskins. But looking at this pattern, here's, I used a circle mark when we collected Entoloma veraskins. Look at this. All these positive yes mark of Entoloma veraskins are all from Chichijima. This time as well, collected from Chichijima, but negative from Hahajima. And each time when we collected Entoloma veraskins from Chichijima Island, fruiting bodies were very abundant. So this pattern is very peculiar. Well, there are always a chance of human biases. You know, even though we went to the island with great mycologists, as a human, we may miss many mushrooms, even though they may be fruiting. So just to show that we were great teams, I show you some examples that we were actually truly great. And one is about the species Boninogaster, named after Bonin Island, Boninogaster. They originally collected from Chichijima Island in 1936, but since then, there are no definite record. So that is a big reason why the Japanese red list lists this species under critically endangered. But ever since I started collecting in the Bonin Islands in 2009, we collect and we keep collecting a lot of floating bodies from almost anywhere in the Bonin Islands, both in Chichijima and Hahajima. So when we try to look for a particular species of mushroom, we can find them. The another example, actually, it's related with Australia. Some of you may know about her, Biz, Elizabeth Sheedy, and together with Hudson, that time the PhD student from Brazil, we visited Bonin Islands together. Then we collected this tiny bird's nest fungi. And this is actually demonstrated to be the Seatus badius, which was actually listed under the extinct category in the Japanese red list, because since early 1900, no collections were made in the islands. So we rediscovered an extinct species from the Bonin Islands with good eyes of those great mycologists. So these are just examples to show you that we were actually able to find mushrooms. So going back to this chart, again, many great mycologists with the equal effort of collecting both in Chichijima and Hahajima. But some people might still say that it could easily be human biases. And I can understand it. All of us know that sometimes during the fieldwork, toward the end of the fieldwork, we get tired and we don't see many mushrooms at the end. Or on the other hand, sometimes at the very beginning of the fieldwork, we were mentally, physically not ready yet. So for the first few days in the field trip, we may not focus so much and we may miss some mushrooms. But again, the sequence of this mushroom survey in the Bonin Islands, sometimes Hahajima first, Chichijima later, Chichijima first, Hahajima later, and so on. So it's not simply that we get so much tired and miss a lot of mushroom at the end of the field trip. And also, we made pretty much equal amount of efforts in both islands. This is just one example from the 2015, and here all dots show that the place we visited in the islands. So pretty much across the island, both in Chichijima and Hahajima. So the equal effort in both islands. The another odd trend is that among the collecting site in Chichijima, which is the only locality of Entoloma veraskins in the Bonin Island system, there are only 3 sites that we successfully collected Entoloma veraskins. And I went back to the original record, specimen record from the other years, and it showed the same thing. We visited many places in the Chichijima, but we successfully collected the species only from 3 sites. So the pattern is becoming really, really interesting and peculiar. So we wonder if that Entoloma veraskins is not distributed in Hahajima at all. So we may wonder there may be it is because of the different phenology, different patterns of fruiting between islands. However, typically speaking, we collect same species of mushroom, both from Chichijima Island and Hahajima Island at the same time. So I don't think the phenology difference is the big factor if that exists. And because that's a very beautiful island, they have ecotourism. Ecotourism is very popular in those island systems. So there are many park rangers and ecotourist guides. So I've interviewed with many people in both Chichijima and Hahajima. But people in Hahajima had no definitive answers about the presence of Entoloma veraskins in Hahajima. So there are no observation record from those islands. And currently, I've been collecting soil samples from both islands. So far, we have detected no DNA of Entoloma virescens from Hahajima. So the species is actually absent from the island. And another distinct trend is that we had absolutely no record of Entoloma virescens from the Bonin Islands, including Shijima Island, since 1854 until 2009. And why is that? So I came up with several naive hypotheses. The one is that the species is so common, too common, that people just ignored. Although it's possible, I don't think it is likely. Because like I explained at the beginning of my talk, the species is rather rare in the mainland of Japan. So if mycologists spend time visiting the islands, it takes 24 hours by ship. And back then it took more than 50 hours and it was expensive. So spending so much time and money and effort, I don't think they intentionally ignore the striking blue entoloma. The other possibility is that they have simply overlooked. But like I said, many skilled, great mycologists have visited islands. I don't think they overlook that rather big blue entoloma from the forest, because they have collected tiny cuff fungi and very small mycena, for example. And I don't think they have a chance to overlook blue entoloma. The other possibility is that there used to be rare fruiters in those islands, but recently becoming very frequent fruiters. I don't know if we can prove that trend, but it's one idea that we have to keep in mind. The last hypothesis is that the species of very frequent fruiters currently in the Tsushima Island is actually introduced from other areas. And if that is correct, that means that the Entoloma Veraskins in 1854 by Wright and Entoloma Veraskins collected by me since 2009 may represent different taxa. And there are some possibility that the species were introduced from the other area. This is one example of ectomycorrhizal mushrooms, Swellus granulatus. It's rather common across many parts of the world, and it is associated with Pinus. But in Okinawa, the other subtropical island in Japan, the species is associated with Pinus lutuensis, that is the endemic species in Okinawa. But like I said, Bonin Island have no known native ectomycorrhizal plants. But currently, Pinus lutuensis was introduced from Okinawa by humans, so that many ectomycorrhizal mushrooms were introduced with those pine. And this picture actually was taken from the Bonin Islands, a very, very common species. Sometimes when we go to planted Pinus trees, Pinus stands in the Bonin Island, we see this species and this species only. There are no other mushroom fruiting. So very, very common introduced species, like Pterotheca hats, also associated with Pinus lutuensis. So the take-home message is that although Bonin Island have no known ectomycorrhizal plants as a native plant flora, currently they have Pinus lutuensis introduced from Okinawa. And it's rather a long history because the first introduction of Pinus lutuensis from Okinawa was made in 1899. So ever since the Pinus lutuensis was introduced to Bonin Islands and established there, many ectomycorrhizal mushrooms associated with Pinus could colonize the Bonin Islands. Before that, because of lack of the ectomycorrhizal partners, they couldn't colonize the islands. And possibly, ever since the Pinus lutuensis introduction was made, the other ectomycorrhizal fungi from other parts of Japan, like mainland, could also disperse and established in the Bonin Islands. So I really believe that the current ectomycorrhizal fungal flora in the Bonin Islands, the mixture of Okinawa, which is probably the major one, and also the mainland Japan, and possibly also from the very South Pacific as well. So the human-mediated introduction of mushrooms is possible, and it is actually true in the Bonin Islands. I'm not saying that the Entoloma veraskins was introduced with pine necessarily because the species is not ectomycorrhizal, but probably the species lack habitat of pine stands. So it is possible that the pine, with the introduction of Pinus liquensis from Okinawa, other mushrooms of non-ectomycorrhizal things like Entoloma veraskins could be introduced from Okinawa. So to understand and clarify this trend, I'm currently studying the DNA barcoding of blue entelomas from many parts of the world. And in the future, I'm hoping that based on the SNP-based population level analysis, I could identify the origin of blue entoloma in the Bonin Islands. But to conclude that, I really need to get the same data from the type originally made by Charles Wright, which is stored in Kew and Harvard. But I keep hearing that those type specimens are in poor quality, very powdery, almost no characters we can see under the microscope. So I'm not sure if you can get good DNA from those powdery specimens. So I'm gonna be finishing my talk very soon, but before the end of my talk, I'm gonna briefly talk about the world distribution of Entoloma veraskins. This is from GBIF, and it shows the scattered records. No specimen records registered yet from Japan, but I have some literature record from Africa, Southeast Asia, Japan, of course, and I have collected myself from New Caledonia. So, it's rather wide distribution. And in New Zealand, we all know that the directly blue entoloma different species exist, and New Caledonia and Japan. And I can't really show you data for now, but my gut feeling is that this entoloma from New Caledonia is more closely related to the Japanese one. I have no proof yet. And during my preparation of this talk, I realized the interesting trend in Australian Entoloma veraskins as well. I got this data from the Atlas for Living Australia, and I showed the distribution record of Entoloma veraskins in the eastern part of Australia. But when I searched the specimen record and also human observation record, it showed very peculiar pattern. That is, the oldest record is from Queensland, stored as specimen in 1912. But ever since then, until 1990, there's no record. I don't know if it is just the incomplete data registration in this system, or actually data are actually lacking, truly lacking. So the pattern seems to be very similar to the Bonin Islands, collected in all time, completely absent of data since then, and recently becoming very common. So my question and homework for all of you is that, are there any specimen record of Entoloma veraskins from Australia between 1930 and 1989? And also, are there any reliable observation records of Entoloma veraskins during this missing period? If it is absolutely lacking during this period, we may have to think about the same hypothesis like I came up for the Bonin Island system. So to think about the taxonomy of Entoloma veraskins, I have to doubt many things, actually. It could be recently introduced from the other island areas, but going back to the Latin name of Entoloma veraskins, and veraskins typically means green. I don't know why the name was chosen, because if I had to describe this species now by myself, I would choose blue or maybe yellow, but not green. It is possible that the blue species with the yellow discoloration makes it greenish looking, but I don't think that is the color that we typically choose to describe the species. And also the original description, Latin, says that it's pale blue becoming green when dried. So when dried, it may be possible, but also says the paleocentral depresso with the depressed center of cap. I don't think that is a typical character for the Entoloma Veraskins. So the materials from 1854, although it was named Entoloma Veraskins, it may not be the same blue Entoloma. And the other scary scenario is this. Was Entoloma Veraskins really collected from the Bonin Island? So you may not understand this comment, but here's the situation. So it was collected during the U.S. Pacific this big exhibition. So it showed the general route of this exhibition. It's very simplified because it doesn't show the Okinawa and the Bonin Islands, but they have stopped there as well. So it was part of the huge exhibitions. And I heard from botanists that some of Charles Wright plant collections had very dubious specimen record. For example, the plants obviously common and only existed in South America were labeled as Southeast Asian specimens, for example. So there are certainly some mixed information records. And the one from Bonin Islands, although the specimen paper slip say so, may not actually have been collected from the Bonin Islands at all. They may be collected from, well, South America, Australia, or somewhere else. So that's a really difficult situation. We have to really make sure that the materials were collected from where. So as very inconclusive conclusions, there are many mushrooms frequently fruit in the Bonin Islands, but they may not be endemic or even native to the islands. Entoloma veraskins, I don't know for sure now, but the current Entoloma frequent fritters in the Chichijima Islands may be introduced from somewhere else. Boninogaster, I don't know, but after I collected in Boninogaster in many places, I have collected in many places in Bonin Island and also outside the Bonin Islands. Entoloma veraskins from the Bonin Islands should represent the true Entoloma veraskins, ideally. However, the true locality may not be the Bonin Islands. So DNA data are great, and I am still trying to get good DNA from Entoloma veraskin specimens from Japan and across the world, but it may be more important to get very basic information like Charles Wright diary and a field notes, just to make sure that he really collected blue Entoloma at the Bonin Islands. So this is a part of the Bonin Islands, a very blue, beautiful ocean, and it's so bad that I can't visit this island due to the pandemic. And hopefully by next year, everything gets back to normal, and we can collect in those beautiful islands again. But during the pandemic, because I couldn't go to fieldwork, I had so much time to do other things. So I was responsible for supervising the Japanese subtitle of this movie, Fantastic Fungi. Some of you may have watched this movie already, but in Japan, this movie starts from this September, and Japanese subtitles are supervised by me. And it was a fun time doing so. So this is the last slide of my talk, and thank you very much for your attention. And I'm looking forward to answer you any questions.
Thank you so much, Kentaro. That was interesting. It seems you've uncovered probably not the first, but a very important scandal in mycology of to the origin of a type specimen. We've already got the first question, and it comes from Bevan. So he wants to know, do you know who first decided that the blue entoloma should be called Entoloma virescens?
Well, like I showed in a slide, it was described by Berkeley and Curtis, and I have to go back to the original description again. But I'm pretty sure they realized that the specimen originally was blue, and ever since then, there are very scattered records, papers and books and stuff like that, and I can't really go back to the comment saying that the Entoloma Veraskins is blue. So that's another thing I really have to study. In addition to the original description, I probably have to search on the literatures in between, after the original description and until 1950 or so. Mm-hmm. So my brief answer is I don't know, but we have to study.
I wonder if it's a case of the person who found it being colorblind.
Well, yeah, that's that. Yeah, we may figure that out. Probably
we have to check the other records.
Right. We need a biography of those people.
That's right. If anyone else has any questions, please pop them in on the question and answer button. You can also raise your hand, and I can allow you to ask them directly to Kentaro. Yeah, I mean, hopefully you'll be able to get out into the field soon. The islands just look spectacular, and I think a lot of us are really craving that kind of field work, no matter where it leads us. So hope you can get out there.
It's so bad that the islands are depending on tourism. Mm-hmm. They have no major other industries, so they are waiting for tourists to come. But at the same time, even though they have about 1,000 people living there, they have no hospital. So once the COVID-19 spread in the islands, it's very difficult to contain. So maybe as a responsible mycologist, I shouldn't really visit now and wait until the things get better.
Yeah, absolutely. Tom May has a question. He's going to ask directly. Tom, just unmute yourself when you're ready.
Hi, Kentaro. Wonderful to see you and hear you. And yes, indeed, it would be marvellous to see you in person sometime in the not too distant future. When you were plotting the records from Australia, you were wondering, why there are no records earlier than, maybe the 1990s. I think it would be useful to plot that against the general density of records for all fungi. And I think it's just that there weren't many records, and a lot of those recent records are through citizen science platforms. So that's facilitated lots and lots of records coming in. So if we look at something like Mycena interrupta, which is probably one of the most kind of obvious and of the macrofungi, that very few herbarium records between the type being collected in 1859 and even the current day. So I think the background level of collecting is just very low. The other interesting thing about the Entoloma virescens in Australia, it's in these very near coastal areas, particularly from kind of the east of Victoria up the coast. So there's, it's not near Melbourne. So a lot of the collections come from near the capital cities. It is quite common in the Sydney area. So I guess that would be the place perhaps to look. But yeah, it's intriguing that there is that gap. But I think in general, there was just a very low base. Yeah, very fascinating to see what's going on. And I look forward to, if you have success with the sequencing, to see what that might tell us.
Yeah, thank you very much for your comment. Yeah, the density of data, that's very important. It may be a lack of mycologists instead of a lack of mushrooms. So that's always a bias that's possible.
Vanessa Ryan actually makes a comment. She says it's also fairly common on the Sunshine Coast, hinterland and Queensland. Bevan, we would like to ask a question too.
Yes, so that previous question wasn't actually from me. But that's cool. It was a good question anyway. Yeah, we've been slammed into a level 4 lockdown here, and so it was supposed to be the meeting room and I sent it around. Anyway, my actual point, yeah, those records of Entoloma virescens from New Zealand, I've just had a quick look at our database, and those are all probably misidentified and really should be Hochsteteri. And certainly it's interesting, you know, when seeing those photos, they are actually really quite different, aren't they? You know, the Hochsteteri doesn't do anywhere near that sort of staining yellow. So I guess my thought process is, well, where else has this happened? And do you have enough access to specimens that you can sequence for your study? Or do you need additional specimens, more help?
Well, within Japan, I think I'm getting enough specimen and data. But outside of Japan, except for 1 or 2 specimens that I collected myself from New Caledonia, I don't have many specimens. And I don't have access to Southeast Asian collections at all. And it would be great to have African collections. But currently I have no data. But I think Australian collection will be great because it's been stored in good condition, I believe, and there are many records. So the first thing I really can do is that to get data, enough data from both Japanese mainland and also Australia and compare. If they are the same, that's great. If not, that's also great. So that's one thing I can compare. But all other areas, Brazil, Southeast Asia, Africa, that's kind of difficult to get.
Absolutely. Genevieve Gates in Tasmania also says that there's actually 2 records from Heathland down there, and she suggests to consider a species complex as well. It does sound like a very interesting conundrum that you've found. So Vanessa Ryan's asking, do the African ones look similar to the Japanese ones?
Yeah. I mean, they're all blue. But like Bevan said, the one in New Zealand, of course, it's called in different names, look different. You know, typically, well, it's kind of difficult to describe how different, but the New Zealand specimen seems to be very different from Japanese ones. But comparing to those 2 species, I think African one and the Brazilian specimens are very similar to Japanese Entoloma Veraskins. So I would say that that's phylogeny true as well. We don't have good DNA data from those areas, but I really believe that the Brazilian specimens and African specimens are closely related to Japanese specimens.
And Genevieve asks if you're doing a multi-gene phylogeny. I suppose that depends on whether or not you're getting some DNA from them.
Well, actually, I didn't show you data for today's talk, but I already have multi-gene data from multiple samples of Entoloma Veraskins. It's not very difficult to do. But like I said, the type specimen become absolutely important. And I don't think we can get reliable multi-gene data from the type specimens of Entoloma Veraskins. So my naive idea originally was that, well, if type specimens are in a poor condition, why not just going back to the type of locality and collect new specimens, which is natural. Many mycologists and many taxonomists do the same thing. But now I'm wondering if that is valid because we don't even know if the type of locality is really the Bonin Islands. So it's really circular.
Specimen, yeah, and whether or not the type specimen is accurate. If there are any more questions, you got a couple more minutes to post any up. Just give it a couple of seconds to see if anyone else is coming up. We've got another question. Hang on a second. Please go for it. Just unmute yourself. Yep, go ahead and ask your question. I'm not sure who exactly that is. It's under Bevan's name, but I've unmuted. All right. Microphone's not working. Feel free to type your question in if you like. Give you a couple of minutes to do that.
So people are learning the, how to use the system, and it's exactly the same in Japan now.
Yeah, even once you know, things go wrong, right? Okay, does your phylogeny show a close relationship with the other blue entolomas? A note from Cook 1879 refers to a green color for one of the species, now plagued in synonymy with Hoxtetori.
No, well, no, I don't have an answer for that, because except for some of my own collection of blue entoloma from Japan and a few other countries, all other names of blue entoloma are not easily accessible for me. And it's very likely that some of them turn out to be a synonym of Entoloma Veraskins, but I don't have any good answer for that. So I really think we should really do the higher level phylogeny of Entolomataceae, including all blue entoloma species, which is not done yet.
Yeah, great. All right. Well, we're nearing the end of our time, so I'm going to start closing out. But yeah, thank you so much, Kentaro, for joining us today, all the way from Japan.
Yeah, well, thank you very much. I enjoy it very much. And I have to tell you that I am currently attending the Japanese Mycological Society meeting now, actually, because it's going throughout this week. So early this morning, I attended part of the session in Japanese Society, and then now I came here. And soon after this seminar series, I'm going back to the Japanese Mycological Society conference. So it's a miracle that we switch between the two Mycological Society conference on the same day across different continents.
You're right. There are certainly benefits to being stuck at home or at least online. So I'm really happy that you could join us today. But also thank you to everyone else for being here to listen to Kentaro's fascinating research. Please make sure you can join us next month. Dr. Yupe Tan from the Queensland Department of Agriculture and Fisheries will be talking about her work with microfungi. And we hope to see you all then. It's on Wednesday, the 29th of September at the same time. But until then, enjoy your day, stay safe, stay sane, and stay sanitized. We'll see you then. Thank you, Kentaro.
Cheers, Kentaro.